Hello, everyone, and welcome to today's live broadcast, New Horizons for Protein Degradation, Immediate, Selective Protein Control using the dTAG system, presented by Dr. Behnam, Nabet, Research Fellow, Dana-Farber Cancer Institute, Laboratory of Nathanael Gray. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Cell Signaling Technology. For more information on our sponsor, please visit cellsignal.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I'd now like to introduce our presenter, Dr. Behnam Nabet. Behnam Nabet is a postdoctoral research fellow in the laboratory of Dr. Nathanael Gray at the Dana-Farber Cancer Institute. Dr. Nabet received his Ph.D. in cancer biology from Northwestern University and a BA in biology from the University of Pennsylvania. In his postdoctoral research, Dr. Nabet developed a generalizable technology platform known as the dTAG system to rapidly degrade any target protein of interest. To read more about Dr. Nabet and the dTAG system, please visit the Biography tab at the top of your screen. Dr. Nabet, you may now begin your presentation.
Okay, great. Well, thank you very much for the kind introduction and for the invitation to present today in today's webinar series. So as was mentioned, I'm a postdoctoral fellow in the laboratory of Nathanael Gray, and the work that I wanted to share with you today, I actually started under the joint mentorship of Jay Bradner and Nathanael prior to Jay's departure to lead Novartis research. Broadly, we're interested in developing chemical tools and strategies to not only inhibit target proteins of interest, but actually send them to the proteasome for degradation. And today I wanted to highlight some of our ongoing work in the development of a technology platform that we call the dTAG system that allows us to degrade any target protein of interest. For many years now, we've been interested in this small molecule known as thalidomide. Thalidomide is quite infamous in part because back in the 1950s, it was used as a treatment for pregnant women to help with their morning sickness. Subsequently, children that were born from pregnant mothers that took thalidomide had very catastrophic consequences. This led to severe birth defects and limb abnormalities that are shown in this image here. A major breakthrough came back in 2010 when Hishiro Honda's lab demonstrated that the cellular target of thalidomide is actually this protein known as cereblon, or CRBN. What the Honda lab showed was that this IMiD could actually bind to cereblon, which is an E3 ubiquitin ligase that's part of the COL4A complex that's depicted here. Subsequently, Bill Kalin's lab, Ben Ebert's lab, and Eric Fisher, prior to his move to Dana-Farber, demonstrated that lenalidomide, an IMiD derivative, has its anti-neoplastic activity by modulating cereblon target recognition. So in this case, when an IMiD binds to cereblon, it also engages what we call a neosubstrate. As a result, this leads to ternary complex formation and the ubiquitination of this neosubstrate and subsequent proteasomal mediated degradation. What these investigators showed was, in fact, that there are many neosubstrates that are engaged upon treatment with an IMiD. Initially, they described Ikaros and Alios, two very important cell type-specific transcription factors that are degraded in multiple myeloma and MDS contexts. This leads to the anti-neoplastic activity of lenalidomide in this context. This was quite exciting to us because in part we thought, well, perhaps since these IMiDs bind to cereblon, we could engage cereblon and direct the cell to degrade a particular target of interest that we wanted the cell to degrade. This led to the hypothesis that we could generate heterobifunctional small molecules where on one end they were hooked up to thalidomide, engaging cereblon, and on the other end hooked up to another small molecule ligand or a binder with high affinity that could engage a target protein of interest. As a result, we could direct the cell to ubiquitinate and then subsequently degrade that particular target protein of interest with this bifunctional molecule. I should note that this idea has been around in the literature for quite some time and highlighted here in this old patent application, indicating that perhaps bifunctional molecules or glue-like molecules that could engage a target protein and the ubiquitination system might actually be able to engineer such a response. So we thought that perhaps this strategy of targeted protein degradation might have many advantages compared to small molecule inhibitors. Over the past several years, we and others have been able to demonstrate, in fact, that some of these hypotheses seem to be correct. We're particularly excited about targeted protein degradation because this allows an investigator to selectively, immediately, and reversibly control a specific target protein of interest. We and others have found that compared to small molecule inhibitors, these degraders can have improved selectivity and prolonged effects, both in vitro as well as in vivo. In addition, these strategies allow one to deplete the entire protein rather than targeting one specific domain, which is a limitation of small molecule inhibitors. What I'll show throughout the presentation as well is that we found that degrading a target of interest actually pairs very well with genetic approaches. These include knockdown or knockout experiments as well. And finally, we are particularly excited about this approach because it opens the door and it gives us an opportunity to tackle very difficult drug targets that have been recalcitrant to small molecule-based approaches in the past. So back in 2015, Jay Bradner's lab demonstrated that they could develop heterobifunctional small molecule degraders of the BET bromodomain family. What Georg Winter and Dennis Buckley, two very talented postdoctoral fellows that were previously in the Bradner lab, demonstrated was that they could take Jun Chi's famous molecule JQ1 that targets BRD2, 3, and 4 and conjugate it to thalidomide. As a result, by generating this molecule that they called DBET1, upon treatment of cells, they could very selectively and rapidly degrade BRD2, 3, and 4. Shown in this Western blot from their paper, what they were able to demonstrate was that really within 1 to 2 hours, they could completely degrade BRD2, 3, and 4. What was particularly striking in this study was not only could this work across multiple different cell lines, but in fact led to improved biologic activity compared to JQ1 alone, both in vitro as well as in vivo. Subsequent to this study, the Gray lab has also been generating heterobifunctional small molecules degraders to target various different kinases as well. One example of this came in Kalle Olson's study back in 2018, when the lab generated this molecule known as thal-SNSO32. What chemists in the group did was take SNSO32, conjugated it to thalidomide in a similar fashion as the DBET1 small molecule that Dennis created. As a result, when Kalle treated cells, she was able to demonstrate very rapid and selective degradation of CDK9 as shown here. Again, within 1 hour of treatment, you could almost completely deplete CDK9. Subsequently, our groups have had quite a lot of success in degrading a number of different target proteins of interest. Many other labs around the world have also been able to use this strategy as well. For us, we focused around bromodomain-containing proteins that are highlighted on the left, as well as various different kinases, including the CDKs. And most recently, we've been trying to degrade aggregated proteins, such as tau, and proteases, such as the HCV protease. So we think that this is really a highly extensible and tractable strategy for depleting targets of interest. Now, when I joined the teams back in late 2015, after Jay's lab had published that initial DBET1 study, what we had noticed over time was that this strategy might have its limitations, one of which is that you need a high-affinity ligand to be able to bind your target protein of interest. As a result, if you're lacking that ligand, you really can't assess whether or not making a small molecule degrader would actually have robust activity towards your target protein. As a result, we had a couple of central questions that I'll try to highlight throughout the presentation with our development of our degradation tag system. What we wanted to do was take a bit of a step back and try to develop a universal tag-based strategy that would allow one to rapidly deplete a target protein of interest. This would allow us to address, on one hand, whether or not a target of interest was worth going after and starting a medicinal chemistry campaign to develop small molecule inhibitors or degraders. It would allow one to evaluate whether or not targeted protein degradation might be an effective strategy for a particular drug target. And finally, we thought that this strategy might have advantages over genetic approaches, whereby, as I mentioned, we could rapidly deplete target proteins of interest within 1 or 2 hours. This could open the door for really investigating rapid biology, particularly in the context of signaling and transcription that I'll highlight today. So this gave rise to our idea that we could perhaps develop a tag-based strategy that we call our degradation tag or our dTAG system. In this particular system, what we do is fuse a target gene of interest to our tag that I'll describe in a moment. We then developed heterobifunctional small molecule degraders that bind that universal tag in a very selective fashion, and then on the other end of the molecule are hooked up to thalidomide, thus engaging cereblon. As a result, this can lead to the ubiquitination of the fusion chimera, leading to its proteasome-mediated degradation. So what I wanted to do today was sort of highlight how we thought about developing this particular system, and then how we've been leveraging it to study oncogenic signaling, which I thought might be of interest to the audience today, how we've been leveraging this to validate and also invalidate different targets of interest, and evaluate rapid biology downstream of transcriptional regulators. And finally, I wanted to end on our efforts to try to evaluate whether or not our system can work not only in cells, but in mouse models. What I hope that you can take away from this webinar today is that our degradation tag system is both rapid and very selective. I'll give highlights on our confirmations on the mechanism of action of the system, and how we've been leveraging it to study fast biology both in cells and our ongoing work in translating this in vivo. What I also wanted to highlight was that there are a number of different tag-based strategies out there in the literature as well. And we initially turned to these and learned a lot from the many papers in which not only describe these technologies, but have used these technologies. Ones that I wanted to just point out as being great solutions that can be paired with our approach as well include the auxin-inducible degron, the halo tag system, the shield system, in particular ligand-induced degradation, and then the SMASH systems as well. So I highly encourage you to take a peek at some of these references that I highlighted here for some of these comparable and compatible strategies. So in getting into the dTAG system, what we decided to do was go after one of the favorite proteins of chemical biologists. This is a protein known as FKBP12. What Gehrig and Dennis had initially shown in their DBET1 paper was not only could they degrade the bromodopain proteins with DBET1, but they also fashioned a second small molecule degrader, which they called DFKBP1, by conjugating SLF to thalidomide. What they were able to demonstrate was that with treatment of cells with DFKBP1, they could degrade endogenous wild-type FKBP12 in a cereblon-dependent fashion. This was quite informative to us because it gave rise to the notion that FKBP12 itself was highly degradable, and we could perhaps degrade this target protein. Next, in looking through the literature, we came across the shield system that Tom Wanless and colleagues at Stanford had developed. Back in 2006, they published a seminal study which they demonstrated that a double mutant version of FKBP12 could be used as a tag. When hooking up this DD domain or destabilizing domain to the target protein of interest, this leads to destabilization of the target. They then describe this small molecule ligand, SHIELD1, which upon treatment of cells leads to the stabilization of this target. This is inherently sort of the reverse of the system that I'll be talking about today, but critically what it demonstrated was that this FKBP12 could be effectively used as a tag. What they also demonstrated with others around the world when they started distributing their technology was that in many different contexts across many different proteins, FKBP12 and these double destabilized mutant versions of it could be used as a tag. So this gave us a lot of confidence that perhaps this could be also a great place to start because FKBP12 could be used as a particular tag. And finally, back in 1998, Tim Claxton and colleagues at Ariad Pharmaceuticals published a bump hole strategy around FKBP12. What they described was a synthetic ligand, AP1867, that selectively can bind to FKBP F36V. This is the particular mutant I'll be focusing on today. This F36V creates a compensatory cavity in FKBP12. As a result, AP1867 can selectively bind to this mutant version of FKBP12 and not wild type. We thought that this would perhaps be a great starting point as we started to develop heterobifunctional small molecule degraders using AP1867 and analogs that we generated of AP1867. With our dTAG system, there are two components. The first of which is one needs to tag their target of interest with mutant FKBP12. We've now done this through the development of lentiviral expression plasmids, which are gateway compatible, allowing you to rapidly clone in your target protein of interest, either with an N-terminal tag or a C-terminal tag. We also commonly include a second smaller tag, such as tandem HA tags that are shown here, which helps in monitoring protein expression if you don't quite have a robust enough antibody for your particular target of interest. We've also used various different knock-in approaches, such as crisp pitch and homology-directed repair that I'll highlight towards the end of the presentation to knock in our tag at an endogenous locus. The second component of our system is the dTAG molecule itself. What I wanted to do throughout the presentation was highlight a little bit about how we came about and landed on dTAG 13 as our particular molecule that we thought was most universal. Here what we did was make an analog of AP1867 that we call ortho AP1867 that's actually more selective than AP1867 itself, that was published by Ariad Pharmaceuticals, that binds mutant FKBP12 and not wild type. We conjugated this ligand to thalidomide to generate the heterobifunctional small molecule degrader. Now, a lot of the biochemical characterization that we initially did in terms of screening and identifying this particular molecule is in the reference that I've cited from my paper that came out last year in 2018. Some of the insights that we gained in our initial study and what we searched for are outlined here on the right in terms of the criteria that we were looking for a heterobifunctional small molecule, dTAG molecule. What we first wanted to do was find a molecule that would selectively bind the mutant version of FKBP12 and not wild type. We didn't want to degrade the wild type protein. We wanted a molecule that could dimerize FKBP12 and cereblon. We also wanted one that would have no off-target activity in the entirety of the proteome, as I'll show. Here we wanted to avoid activity on the cereblon neosubstrates that I mentioned, such as Ikaros and Aleos, or IKZF1 and 3, as well as wild type FKBP. We also wanted to ensure that our molecule works through the defined mechanism of action. As I'll show, we wanted to ensure that the molecule requires cereblon, as well as the proteasome machinery to degrade a target of interest. And finally, we wanted a small molecule that would induce both acute and prolonged degradation of multiple different targets across various different cell lines that had the potential for working in vivo. So collectively, these are some of the criteria that we used initially in a generation of about 65 different molecules that we evaluated and landed on dTAG 13, which will be the focus of my discussion today. Now, skipping over some of the biochemical data that I mentioned that you can find in my manuscript, what we set up initially was a dual luciferase assay to identify small molecules that actually penetrate the cells and could lead to degradation selectively of an FKBP12 F36V fusion protein. And so here what we did was hook up wild-type FKBP12 and mutant FKBP12 to nanoluciferase. As a result, this would allow us to use non-invasive bioluminescence measurements to evaluate whether or not we could degrade a particular target of interest, here focusing just on FKBP12 itself. What we were able to evaluate here was selective degradation of wild-type FKBP12 versus mutant FKBP12. What we found here, as exemplified with dTAG13, that we had really nice dose-dependent degradation of mutant FKBP12 that was fused to this nanoluciferase, and no activity on wild-type FKBP12. At higher doses, we saw the characteristic hook effect, in which you have saturation of mutant FKBP12 as well as cereblon. As a result, you fail to form the ternary complex and can't degrade the fusion. Building on this work, then we could confirm this by Western blotting, demonstrating that 10 to 100 nanomolar dTAG13 treatment in cells that had cereblon intact would lead to the degradation of mutant FKBP12. We could rescue these effects when we knocked out cereblon, as shown here on the right. This demonstrated to us that, in fact, cereblon was required for these effects. Now, subsequently, after identifying a molecule that could actually penetrate cells, we decided to see if we could actually degrade fusion proteins where we fused mutant FKBP12 to a particular target of interest. As I mentioned, we developed lentiviral plasmids where one could fuse either with an N-terminal tag or a C-terminal tag. Here, we started with BRD4, which we cloned into our system and expressed in MV411 cells. What we were pleased to find was that upon treatment of dTAG13, within 1 hour we could see effective degradation of BRD4. This lasted up to 24 hours and also actually up to many days in this experiment. Subsequently, we were quite excited, so we started tagging a lot of different target proteins of interest that we were studying in the lab. These include MYC, EZH2, HDAC1, PLK1, and mutant KRAS. These are either cytoplasmic or nuclear proteins, kinases or transcriptional regulators or signaling effectors. What we found in these MV411 cells was in each case we could nicely, rapidly degrade each target fusion protein, here with evidence of degradation within 1 hour of treatment. What we found was that mutant KRAS was actually the slowest to degrade, where we saw degradation occurring within 4 to 8 hours after treatment. Now from here, what I wanted to do was shift focus and highlight some of our work that we've done in further characterizing mutant KRAS with our system. I think this example helps illustrate how we demonstrated the selectivity of our system and how we can use it to study rapid biology downstream of oncogenic signaling. So as a quick overview, we and others around the world have been interested in trying to develop strategies to target mutant KRAS. The RAS family are signaling effectors that coordinate many different signaling cascades, including the MEK, ERK, PI3 kinase, or all GDS pathways. They're essential for promoting cell proliferation upon mutation, leading to cell transformation. The RAS family is the most highly mutated oncogene family, and in particular, KRAS is the driving lesion in many different cancers, particularly in pancreatic cancer, which is one of our major focuses. Currently, the RAS family is largely very difficult to drug. There's a lot of really exciting new strategies harnessing covalent inhibitors to target KRAS G12C. However, in pancreatic cancer, G12D and G12V are the most common mutations, and still effective strategies to target this oncogene is necessary. Over decades of work have focused around targeting mutant KRAS directly as well as indirectly. And so today I wanted to highlight using our dTAG system how degradation might be a useful strategy for targeting mutant KRAS directly. And secondly, what I wanted to highlight was an alternative strategy that we and others have been employing to identify synthetic lethal interactors with inhibitors that target MAP kinase signaling. And what I wanted to do was highlight a more recent study in which we've identified a synthetic lethal interactor with MEK inhibition, which we were able to validate with the dTAG system. So starting with mutant KRAS, what we decided to do was N-terminally tag mutant KRAS and first demonstrate that our fusion oncoprotein was functional. We turned to NIH3T3 cells, which are a mouse fibroblast cell line that has been commonly used by many investigators over decades of work to evaluate the functions and activities of mutant KRAS. What we observed in line with the literature was, in fact, that our fusion chimera was functional. This led to elevated phosphorylation of MEK, as well as ERK signaling, as well as phosphorylation of AKT shown in the DMSO control lane. Subsequently, upon treatment with our dTAG13 molecule, what we observed was that we could see rapid degradation being triggered within 1 hour of treatment. We saw about 50% degradation of RAS. By 4 to 8 hours, we saw about 90 to 95% degradation of RAS, in line with actually our MV411 experiment, the human leukemia line. What was particularly striking to us in this experiment was actually looking at the 1 hour time point in which phosphosignaling actually collapsed right back to baseline levels within 1 hour of treatment. Here, phosphorylation of MEK and phosphorylation of AKT looks indistinguishable from the control cells. Importantly, treatment of our control cells, which are mock transduced cells, leads to no effects on phosphorylation of these signaling effectors as well. We could also do a number of different control experiments to ensure that this was in line with the proposed mechanism of action of our small molecule degraders. We could knock out cereblon, as I showed earlier, and rescue the effects. We could also block the proteasome with carfilzomib. This rescued the effective degradation of mutant KRAS. We could block netylation, which is required for the activation of E3 ligases with MLN4924. And we could also saturate cereblon with lenalidomide. We also characterized extensively the biology of these cells. What we noticed just looking at the cells was that when we put in our functional mutant KRAS, this led to characteristic changes in the morphology of these cells compared to control. What was particularly striking was that within 24 hours of treatment, we could flip the cells back to looking exactly like the control cells. We could also quantify some of the changes in proliferation and transformation. And so I'm just highlighting here one experiment that we did where we demonstrated that when we expressed mutant KRAS in these cells, we had increased cells in S phase. Upon degradation within 24 to 48 hours, the cells began cycling exactly like the control cells. Subsequently, with the system, this allowed us to really hone in and evaluate the selectivity of our degrader and also evaluate whether or not we were getting the expected effects from degrading mutant KRAS. We turned to quantitative mass spec proteomics in part to first evaluate whether or not we had selective degradation of mutant KRAS. Here we did temporal profiling, and I'm showing here just a 1 hour treatment. We also did a 4 hour treatment, which is included in my manuscript. What we observed at this initial time point was that mutant KRAS and the fusion chimera was actually the only significantly degraded target protein in the cell. We didn't hit FKBP12 wild type as we expected, nor did we degrade any other targets. We also coupled this with a phosphoproteomics experiment to confirm our Western blot findings where we were evaluating phosphorylation of MEK, ERK, and AKT. What we confirmed was that ERK1 and ERK2 were the most profoundly dephosphorylated target proteins of interest. In addition, we identified a number of other different signaling molecules that we've been following up on. And finally, we coupled this with transcriptional profiling to look to see how mRNA levels were changing in the cell upon KRAS degradation. In this experiment, I also compared this to trametinib, a MEK inhibitor. What we observed was that within 4 hours and then also at 8 hours, we could really robustly flip the aberrant transcriptional program that's coordinated by mutant KRAS. We also saw these same effects when we treated with trametinib. This gave us insight into the fact that, as expected, the target genes that were modulated upon KRAS loss are largely ones that were coordinated by MEK-ERK signaling as well. So overall, collectively, these studies helped us not only hone in and evaluate the selectivity and the mechanism of action of our dTAG system, but also allowed us to start to demonstrate that perhaps targeted degradation of this fusion oncogene, and in particular G12V itself, might actually be a very attractive strategy for trying to target mutant KRAS. Now I wanted to switch gears a little bit and talk about the second idea that I mentioned in terms of identifying potential synthetic lethal interactors. What's become quite clear with small molecules such as trametinib and many other MEK inhibitors that are currently used in the clinic is that they fail to produce the expected results in KRAS mutant context. As a result, we and others have been searching for ways to sensitize cells, in particular, and hopefully patients, to MEK inhibition by degrading or inhibiting a second target. So this study was done in collaboration with Bill Hahn and Andrew Aguirre's lab here at Dana-Farber Cancer Institute. And what the Hahn and Aguirre labs had been doing for quite some time are genome-scale CRISPR-Cas9 screens to identify selective dependencies in various different contexts. In this particular study, what they set out to do was treat cells, and these are KRAS mutant pancreatic or lung cancer cells, and I'm showing a snapshot from one of the pancreatic cell lines that I'll be highlighting, in which they treated these cells that are fairly resistant to MEK inhibition, and then perform the CRISPR screen. What they identified over and over again were not only components of MAP kinase signaling that were synthetic lethal interactors, such as MAP kinase 1, SHP2, and other different targets that were expected, they also identified a very, very interesting scaffolding protein known as SHOC2. And this came up as the most highly recurrent synthetic lethal interactor that would sensitize cells to MEK inhibition. Now, a little bit of quick background on SHOC2. SHOC2 binds directly to MRAS and complexes with PP1C. This complex actually regulates serine 259 phosphorylation, which is an inhibitory event on RAF kinase. Upon serine 259 phosphorylation, this actually abrogates the activity of RAF itself. So overall, what the Han and Aguirre labs had been able to demonstrate after doing their CRISPR screens was to functionally validate and evaluate SHOC2 using genetic approaches. We then teamed up to tag SHOC2 in these pancreatic cancer cells and knock out the endogenous locus, creating isogenic cell lines that would give us tunable control over SHOC2. This would allow us to do chemical degradation experiments to evaluate whether or not SHOC2 loss in combination with MEK inhibition actually was a potential strategy that one could pursue further. Here in these pancreatic cells at a 50 nanomolar dose of dTAG13, what we could confirm was very nice degradation of SHOC2, evident within 30 minutes of treatment and near maximal by 1 hour of treatment, in line with a lot of the kinetics that we've been seeing with our approach. As a result, this allowed us to do combination experiments. What their labs had been able to demonstrate and confirm from the literature was, in fact, these cells are quite resistant to MEK inhibition. So here, just comparing lanes 1, 2, and 3, what's quite clear is that upon trametinib treatment, the cells respond very quickly, and there's a loss in phosphorylation of ERK. However, within 24 hours of treatment, phosphorylation of ERK levels return. So here, we were able to ask the question now, if SHOC2 was in fact mediating and was important in this recovery of phosphorylation of ERK, does degradation of SHOC2 lead to abrogation of this effect? And what we observed by degrading SHOC2 was really pronounced maintained loss of phosphorylation of ERK. We could also confirm then in this temporal profiling experiment that we saw reactivation of phosphorylation of CRAF. As I mentioned, this is an inhibitory phosphorylation mark, as a result, explaining in part this effect on phosphorylation of ERK. As a result, this was able to confirm some of the interesting observations that they had had in their genetic experiments. Those acute experiments could then be extended to more prolonged proliferation experiments. So we could ask the question of whether or not combining MEK inhibition with SHOC2 degradation could in fact impair proliferation. What we had been able to confirm was that trametinib treatment upon SHOC2 deletion leads to a very pronounced effect. This could be rescued upon adding back SHOC2 wild type, or our fusion chimera, the SHOC2 FKBP12 fusion protein. What we had observed was in fact that SHOC2 itself is dispensable for proliferation of these cells without any perturbation. We could confirm this not only with SHOC2 knockout, but then with dTAG-mediated degradation. And finally, then we could do the real experiment where we could then combine SHOC2 degradation with trametinib treatment. And what we observed was that we could have a pronounced effect on the viability of these cells upon degradation of SHOC2, really confirming this observation that SHOC2 loss in combination with MEK inhibition might be a potential strategy that we should pursue in the context of using MEK inhibitors in KRAS mutant contexts. Now, moving on a bit from some of these signaling experiments that I wanted to highlight today, I wanted to give out some references from some of our other work that might help illustrate really where I see some of the power of the dTAG system. Our first demonstration of the utility of dTAG is shown in this publication here, led by Michael Erb, a former graduate student in Jay Bradner's lab. What we were able to demonstrate and initially identify was this component of the super elongation complex known as ENL was actually a very important cell type specific dependency. CRISPR-wide, genome-wide CRISPR screens were able to identify ENL as a selective dependency. Because ENL was so important to these cells, the only way that we could really study it was by generating isogenic dTAG lines. Here we were able to tag ENL using our lentiviral system and knock out the endogenous locus. This again gave us nice tunable control over ENL and allowed us to do a number of different transcriptional and epigenomic profiling studies to really define the mechanism of action of ENL. This was a nice example where we could go from CRISPR screens to degradation-based experiments, and also were important in our validation of this particular target as a dependency in AML. In concert with this study, we were actually also studying a kinase known as MELK in the lab, which was thought to be very important in triple-negative breast cancer cells. Hubert Huang, a former graduate student in Nathanael's lab, had been developing small molecule inhibitors to target MELK. However, these inhibitors were having really little to no activity when he was treating these breast cancer cells. We decided to take a step back, performed a number of genetic experiments, and also created some dTAG systems where we were able to tag MELK, knock out the endogenous locus, giving us, again, tunable control over MELK. Overall, what we were able to observe was, in fact, that MELK seemed to be actually dispensable for proliferation of triple-negative breast cancer cells. And so in this case, our study ended up being one where we sought to invalidate MELK as a kinase that was important in this context. We've also been able to use knock-in approaches to knock in our tag at endogenous loci. One of our first examples here was we were able to knock in our tag at the BRD4 locus. This gave us an opportunity to selectively degrade BRD4 with our dTAG molecule and compare this to our pan-BET bromodomain degraders, dBET1 and our second generation dBET6, which, as I mentioned, hit BRD2, 3, and 4. So this was a nice example where we could then compare the effects of selective degradation versus pan-degradation of this important family. We also teamed up with Rick Young's lab in a study led by Abe Weintraub, in which we had been investigating a transcription factor known as YY1. Here again, we were able to use knock-in approaches to tag YY1 and selectively degrade YY1. This allowed us to study how YY1 regulates enhancer promoter loop formation as well as transcription in general. And finally, more recently, we teamed up with Margaret Gradell's lab at Baylor and were able to tag a mutant oncoprotein known as NPM1. Lorenzo Brunetti and Michael Gundry, two leaders of this study, together we were able to demonstrate that we could effectively degrade NPM1 by selectively tagging the mutant version of NPM1, and then study its effects on transcription and identify the HOX cluster as key essential target genes downstream of NPM1. So hopefully some of these references that you can turn to in the future will help illustrate how we've been leveraging the dTAG system to study transcriptional regulation, in part with ENL, BRD4, YY1, and NPM1, and how I think it's been valuable to us to not only validate targets of interest, but is an important step for us to also invalidate targets of interest and decide whether or not a medicinal chemistry campaign is actually worth the investment of funds and resources. And the final piece of data that I wanted to highlight today was our ongoing effort to see whether or not we could develop a system that had the potential to work in vivo. And what we decided to do here was generate a fusion chimera that would allow us to do non-invasive bioluminescence measurements in vivo. And what we did was tag luciferase and express the luciferase FKBP12 fusion in MV411 cells. Here is an in vitro experiment demonstrating that we could effectively deplete luminescent signal upon treatment with dTAG13. We then set up the following experiment. We tail vein injected these MV411 cells into mice and let them engraft and monitored their leukemic burden. Subsequently, on the first day of the experiment, we evaluated their baseline luminescent signal. The next 3 days, we performed treatments in the morning. After days 2 and day 3, we then performed again bioluminescence measurements. This allowed us to capture potential effects on luciferase signal. On the final day, I didn't perform the treatment in the morning, and we just did the luminescence measurement to see how this affected, to see the recovery levels in response to breakdown and washout of our compound in vivo. So here's what the data looked like from some of our mice, comparing 3 of our vehicle versus 3 of our dTAG13 treated mice. At baseline, they had relatively similar leukemic burden as exemplified by their signal. By day 3, which was where we saw the most pronounced changes, we can compare the vehicle control to our dTAG13 treated mice. here we saw significant repression of signal. Upon evaluation day 4, which was 28 hours after our final treatment, what we saw was recovery of signal, illustrating the reversibility of our particular system. And here's the quantified data, just showing the various different time points, where we saw really pronounced effects both at day 2 and day 3. These are 4 hours after treatments where we did the non-invasive measurements, and then the recovery and the reversibility of the system on the final day. And so this was quite an exciting observation for us because it was able to provide our first demonstration that perhaps our system had the ability to work in vivo and degrade targets in vivo. And so with that, I'd like to start to wrap up. What I hope I was able to highlight today was that the dTAG system that we've developed is able to induce potent, immediate, and selective degradation both in cell lines and in mouse models. Our system enables both target validation and invalidation with some of the examples that I noted. I think it's particularly powerful for studying transcriptional regulation, these rapid changes in transcriptional signaling that can occur upon loss of a transcription factor or a transcriptional regulator, with some of the studies that we've now published that I tried to highlight. I also wanted to note that I think this system works quite well for looking at and evaluating rapid changes in signaling, as hopefully I was able to illustrate with our studies and our ongoing work in evaluating mutant KRAS and how targeted degradation of mutant KRAS, particularly the G12V mutation, can collapse both oncogenic signaling as well as transcription upon acute loss of KRAS, and this is able to lead to the reversal of the aberrant biology of these mutant KRAS expressing cells. And finally, some of our ongoing work in terms of combining CRISPR screens and evaluating synthetic lethal interactors with various different inhibitors, as an example with the MEK inhibitor trametinib, and our identification of SHOC2 as a key scaffolding protein that might be a modifier of sensitivity to these inhibitors. And so with that, I wanted to thank a number of people. First and foremost, I need to thank my mentors, Nathanael Gray and Jay Bradner, really for their continued mentorship and support over the past several years. They've been really outstanding mentors, and I can't thank them enough. I also need to highlight 2 of my teammates, Dennis Buckley and Justin Roberts, in our initial description and demonstration and development of the dTAG system. They were really great friends and colleagues to work with. I also need to thank the Gray Lab for being such an awesome team to continue to work with. Hopefully I was able to highlight our various collaborators that I've been working with over the years, Rick and Abe, Kwok and Anand, Peggy, in particular Lorenzo and Michael, Bill Hahn, Rita, Jason, as well as Andy as well. All these collaborators have been instrumental in terms of our understanding and evaluation of the dTAG system. And finally, I wanted to acknowledge my funding from the American Cancer Society, the BAR program, and the Pinard Fund, as well as Nathanael's funding that has helped support the work from the Hale Research Center, as well as the Pinard Fund as well. And finally, what I wanted to note was that I've really tried to make our system as open and as accessible as possible for interested investigators to try to use. We're happy to share any reagents or insights or ideas that might be helpful for various investigators' ongoing work. We've tried to set up a rapid express MTA that allows us to share our molecules with interested investigators. Our plasmids and reagents are all on AdGene, where you can find also protocols and some guidelines. And if you contact me, I'm also happy to direct you to some other additional resources or send you them myself as well. And it's been great to have the opportunity to really try to, in an open source fashion, try to distribute to as many interested investigators as possible around the world. And so we're really excited to see how the dTAG system performs and how this might actually help push our understanding of fundamental biology as well as application towards treatments of various diseases forward. So again, I wanted to end by thanking the organizers for the invitation, for putting this all together, and I'm happy to also take any questions as well. Thank you.
And thank you, Dr. Nabet, for this excellent presentation. We've got lots of questions coming in, and let's get started on these. I want to remind our audience that if you do have any questions you would like to ask, now's the time to do it. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. So let's jump right in. Our first question, in vivo, would you expect side effects of inhibiting major proteins?
I think, yes, that's definitely true. In general, I think that's quite a concern when you're trying to target an endogenous protein, either with a small molecule inhibitor or with a small molecule degrader. For our tag-based system, the great part about trying to degrade on target is actually that our molecule is so selective that the only thing that we degrade is what's fused to our tag. So we can really get cell type specific degradation of that particular target, which really helps us evaluate what the consequences of loss of that target of interest is with the dTAG system. However, in general, that's definitely a broader concern when using any kind of inhibitor or small molecule degrader.
Thank you, Dr. Nabet. Our next question, how does dTAG compare to other tag-based degradation strategies like haloprotag?
That's a great question. So I think our strategy is very comparable to a lot of these different tag-based strategies. It operates in a bit of a different mechanism. I think the most comparable strategy indeed is the haloprotag approach developed by Craig Cruz and colleagues at Yale. One of the key differences is that their strategy works through a covalent mechanism, so the chloroalkane, which binds to their halo tag, is quite different. That might have some potential disadvantages when you think about how degradation and the kinetics of degradation, as well as the reversibility of degradation, might operate. In terms of comparisons to other strategies, I think one key advantage here is that we're hijacking cereblon rather than expressing a second component being the E3 ligase machinery, such as the auxin-inducible system, and that's kind of required for that system to work. And I think a key distinguishing feature right now is that our strategy, as far as I'm aware, is the only one that really has the potential for working in vivo. So, but overall, I think they're all excellent strategies that one can also combine with our strategy as well, perhaps to degrade 2 or more targets of interest. So it'll be interesting to see how investigators kind of also figure out ways to combine our approach with theirs as well.
Thank you. Our next question, how transferable are insights from dTAG to PROTAC design?
Yeah, thanks for the question. I think that this is one where it's to be seen. We're trying to evaluate how this might help us guide development of PROTACs or heterobifunctional degraders with our particular system. I think on the one hand, what's most valuable is just for evaluating, before making the degrader molecule, whether or not using dTAG you can actually effectively degrade the target protein of interest and then elicit the expected biology that you want to see. So whether or not you get the expected effects on transcription and signaling and proliferation and so on. So really as a mechanism to validate that target. I think a limitation here and a concern would be that it's still to be seen whether or not this approach can actually predict whether a bifunctional molecule can be made for that particular target of interest, in part because we've seen examples where both the tag and the target get ubiquitinated in response. And so when you're targeting the endogenous protein, you won't have the tag there to get the ubiquitination on that tag. So if that's the driving event, then you might have a more limited scope of being able to identify whether or not this is very predictive of a PROTAC itself. But overall, as a tool for target validation and just evaluating degradation, I think it's a key platform to be able to do that.
Thank you, Dr. Nabet. Now, have you seen any cases in which there are differences in results from genetic knockdown versus dTAG-mediated degradation?
Thanks for the question. I think overall, we do see a really high concordance, as I showed throughout the presentation, between genetic perturbations and then dTAG-mediated degradation. I think one of the key advantages that we have is really the kinetics of degradation that you get. So you can start to do your experiments within 1 hour of treatment rather than waiting many days after you try to do a knockdown experiment or try to isolate single-cell clones in the CRISPR case. And so as a result, I think it pairs very well with genetic approaches. It does have a particular advantage after you've built the system. But overall, we do see really nice concordance between the two. We see striking differences between trying to inhibit targets of interest and then degrading them. And so I think that is where we are beginning to see really profound potential differences. But overall, with these loss of function approaches, it's a great way of being able to do that more rapidly.
Thank you for that. Now we have some great questions coming in. We have time for a few more. I do want to remind our audience that those questions we do not have time for today, and those that come in during our on-demand period, they will be answered via the email address that you provide at your time of registration. So let's continue on with a couple more questions here. Dr. Nabet, do you see a range of kinetics of degradation with dTAG?
Yes, thanks for the question. We definitely do see a range of kinetics. I think overall, most targets of interest when the system is working very robustly, you do see degradation, as I showed, within 30 minutes to an hour. There are some cases, like mutant KRAS, where the degradation takes a bit longer, so you can see evidence of degradation within 1 hour of treatment. However, it takes 4 to 8 hours really to get kind of that maximal 90 to 100% degradation. What's quite interesting is then really once you're triggering the degradation, sometimes you're able to see in those slower cases the beginnings of sort of those immediate effects of losing that target. So in the example of RAS, we only saw 50% degradation within 1 hour, but complete reversal of the aberrant oncogenic signaling program within an hour of treatment. But overall, I think in general, between 30 minutes to 1 hour, you do see a nice degradation of your target of interest.
Great, thank you. Our next question, is it possible to have different outcomes from the inhibition of the same protein?
Yeah, so I think this question is getting at a key question kind of in the degrader field as to whether or not targeted inhibition can elicit different outcomes compared to targeted degradation. And I think that that's definitely true. I think there are examples in the field now that are harnessing this idea where depleting the target and getting rid of all of its activities are actually quite different from killing the activity of a particular target of interest, such as a kinase. So a kinase, you might be able to have a small molecule inhibitor that affects its kinase activities. However, the protein might have various different domains that can bind to other effectors. It can function as a scaffolding protein. It might be localized in different compartments in the cell. And so just inhibiting the kinase activity might not be sufficient in eliciting the expected response. So we've seen cases then when you can deplete that target of interest using dTAG or an endogenous degrader, you can really have different and more pronounced effects. And so I think overall, those are the cases that we kind of search for where we can see advantages of degradation versus small molecule inhibition.
Thank you. Next question. When cereblon is hijacked by the dTAG, what happens to the endogenous substrate of cereblon?
Yeah, thanks for that question. So that's something that we were really concerned about and we monitored very closely from the beginning of our studies. And so a lot of these kinds of control experiments are detailed in some of our supplemental figures. But overall, what we tried to ensure with our dTAG molecules is that we aren't affecting any of the substrates of cereblon. And so the proteomics experiments are ones where we are able to really assess that and try to evaluate whether or not we're hitting any of those targets. So for example, what we closely monitor are always our Icaros and Alios casein kinase, Sol4, and some of these other neosubstrates that are engaged by the IMiD portion. And we have confirmed that our molecules, particularly molecules like dTAG13, do not hit any of those neosubstrates. I think in general, some of those cereblon neosubstrates are still being identified, and we're continuing to kind of monitor the field and evaluate them. But the proteomics experiments really allows us to identify those in an unbiased fashion, and our molecules do not hit any of those. So really, you get selective degradation of only your fusion chimera.
Thank you. We have time for a few more questions. Let's squeeze some in. How reversible is the dTAG system?
Great. Yeah, so definitely an advantage that I kind of glossed over with all of these degradation-based approaches, as well as a dTAG system, are that they can be quite tunable and reversible. And so one can do washout experiments in culture to be able to then temporally evaluate recovery of the target of interest, and then see if you can reverse the effects when the target comes back. So we've done this, and it's in the supplement of our initial dTAG paper that came out last year, where we did these kinds of experiments, degrading mutant KRAS and then washing out dTAG 13, and looking at the kinetics of recovery, and then kinetics of recovery of signaling as well. So one can wash out compound, let target come back, treat again, degrade target. And so those are some nice applications of being able to use the system as well.
Excellent. Now, this question's come in a couple times. Does the system work in mouse and human cells?
Great. Yeah, thanks for the question. So it does. And so that's why, in part, I tried to kind of highlight some of that, some of those notes in the slides as well. And I'm happy if there are more questions about specific ones that we have also tested in addition to the ones that I noted throughout the presentation. In terms of mouse cells, those NIH3T3s are mouse cells. We've done this in mouse embryonic cells extensively as well, so it works great in those kinds of formats. Most commonly, we've done this in human cells, in part because we're studying human cancer and developing using human cell line models, and it really works great in human lines as well. So you're able to co-opt both mouse and human cereblon with the dTAG system.
Sorry about that. Can you degrade membrane-bound target proteins with dTAG?
Yeah, great question. So we are actively working on that, and it's something that we can do. So we've mostly focused around cytoplasmic and nuclear factors just based on our kind of research interests. But overall, we can confirm that we've been able to degrade membrane-bound proteins. And in particular, when mutant KRAS is active in the cell, it does translocate to the plasma membrane as well in order to elicit its oncogenic effects. So we do know that we can degrade proteins in various different compartments, including the membrane as well.
Thank you, Dr. Nabet. And let's wrap with this final question. What is the spectrum of cell lines that have been used for dTAG studies? Yeah,
great question. So as I mentioned, we've done this in fibroblasts, in human and mouse embryonic stem cells as examples. And then a lot of different cancer cell lines are what we focused on. So we've been able to achieve this in breast cancer cells, in leukemia and lymphoma models, breast cancer cells, and many other both solid and liquid tumor models as well. And so I think that it seems to be quite tractable as you move across systems, in part because cereblon is pretty ubiquitously expressed. And so as long as you have sufficient expression levels in your context of interest, it's worth starting to evaluate if you're interested in those particular contexts.
Dr. Nabet, thank you so much for this excellent presentation and this great interactive Q&A. Do you have any final comments you'd like to leave our audience?
Oh, I appreciate that. So I really want to, again, thank the organizers for putting this together. Hopefully it was useful. As I mentioned on my last slide, you can contact me through direct message or tweet at me, and I'm happy to further share reagents, insights, collaborate, and help you try out our system for degrading your targets of interest. So thanks a lot for your time.
Thank you again. And before we go, I would like to thank our audience for joining us today and for their interesting questions. One final reminder, those questions we did not have time to get to today and those that are submitted during our on-demand period will be addressed by Dr. Nabet via the email address you provided at the time of registration. I would like to one more time thank Dr. Behnam Nabet for his time today and his important research. I'd also like to thank LabRoots and our sponsor, Cell Signaling Technology, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. We thank you for joining us, and we hope to see you again soon. Have a great day.